So this is a little different than a moisture balance, a moisture analyzer. Tools that come out seen before the past 20, 25 years. We at Aspen use the ContiTrack Vapor Pro made by Arizona Instruments. There are other manufacturers, but this is the one we have. That's what I'm going to talk about. A sample is weighed into a vial and sealed in a vial with the cap of the septum. And then the septum, the vial is put in a machine, pulled inside a heating block where the septum is punctured by a needle. And the needle is actually, it's a hollow tube with another tube within it. So you have gas going inside the tube, inside of the first needle, coming out to the void, to the headspace of the vial, and then pushing anything in the headspace back into the block where it goes past a humidity sensor that measures the moisture. And this does away with a lot of interferences from other volatiles because the humidity sensors are fairly specific to water. There are some things that will be interference. One of the things we found fine particles, we do some work with a powder activated carbon where they carried the sample, went into the block in the vial, and the first step is to purge the vial. Well, during that purge step, you fluffed up a bunch of this carbon, you drew it back in the needle into the block, and jammed the FRIT on the humidity sensor. So we never got an impact. Also, if you're dealing with something like a shredded plastic light can get stuck in the holes in the needle, that happens, you plug the needle, plug the flow. In those instances, we unscrew the needle, take it apart, and just hold the needle under the torch and burn it out. It is a versatile instrument. Yeah.
Sample size, do you rate it based on how much water you're driving out?
Yeah. Yeah. We can go anywhere. Some of the lower moisture samples, we use like 2 or 3 grams. Because you want to see a certain amount of water going past.
How much do you really want to see?
I believe these things can seep up to gram quantities of water. It just gets to the point where you're generating this water, the sensor's seeing it, and depending on how you set your end conditions, whether you want to set at a certain, if the water coming off the sample drops below a certain rate, consider it done, or for a certain period of time. But if you're doing like a rate type endpoint, where your sample slowly leaching out water, you don't want to be waiting 3 hours. So it's all in your method development. It's an easily calibrated and verified instrument. Our method is you calibrate the sensor itself, so for traceability, verification. And it's a very user-friendly instrument. We'll have one out here, and we'll be using one of the methods we often use here at Aspen. It's kind of anticlimactic. You weigh a sample, put it in the machine, push a couple of buttons, then you wait, and step back and wait for the data, which is fine. Well, we can get is you get these little capillary glass tubes that are designed to contain a finite amount of water. Take the tube, touch it to it, drop of water in it, sets up a known volume of water, and then you put that in the cell, and then there's a sample, there's a routine where it knows what the microprocessor knows how much water should be seen, and then it will adjust itself. The last method I'll talk about, we have a analyzer set up on top of that. It's a Karl-Fischer method. This is a chemical method. Chemist? Orange here? All right, good guys. This is a chemical method for measuring water content. Two popular routes to Karl-Fischer, volumetric titration, or most often used to use here is the volumetric titration. The difference between that. It is fairly specific to water. It's very sensitive as well. You get able to measure micrograms of water in a sample, all the way up to, I've done some samples 80–90% water, just because of the nature of the sample. I couldn't use a lot of weight loss drawing method or moisturizer, so I went with Karl-Fischer. It's not restricted to any one sample form. Solid liquids or gaseous samples. It does involve, however, the use of non-aqueous reagent because it's the sensitive moisture method. Even water vapor within the cell will cause your endpoint to drift and give you give you false high results. So there's a fair amount of lab technique involved. Okay, it's not difficult, but you have to pay attention to the details. You have to also have to go into the sample. Some samples, depending on your composition, you have a sample that's rich in aldehydes or ketones. That can cause side reactions. That will give a steadily phasing endpoint, but the reagent manufacturers have accounted for that, use by special reagents in the cell, and all will be right. Also pH, if you have a highly basic sample, there are side reactions that can occur within the cell that cause the endpoint to fade as it's reached. Also, if your sample pH is too low, the reactivity constant of the reaction drops, and you get a really slow, sluggish response. So the sweet spot is about a 5 to a 7 pH within the cell. And most of the reagents you purchase have a fairly decent buffering capacity, and you are using somewhat smaller samples, so that's what you usually account for. But again, you have to do your due diligence in your method development. You do have to maintain the equipment. You are dealing with electrodes, sealed cells. You have to make sure all of your seals are vapor tight. And sampling reduction, usually use syringes, so you have to make sure you're sampling right. For the chemist in the room, put up a couple of equations. Basically, how this reaction occurs is that you have methanol is typically the solvent of choice for Karl-Fischer. There's a certain amount of SO2 in the form of SO3 in solution and iodine present. And whenever water is introduced to the sample, the reaction can proceed in this direction. And as long as there's water in the sample, this reaction can proceed to the right. And as soon as the last bit of water is depleted, an excess of iodine is detected by the electrode, which is just a two-pronged vacuum probe measuring the potential in the solution. First hint of free iodine, potential drops. The microprocessor sees that as the endpoint, stops the titration, gives you your data. So you get a stoichiometric 1-to-1 molar reaction with the water with the iodine in the presence of SO2, or actually sulfite in solution. It reduces the iodine to iodide. Again, the first excess of iodine in solution is your endpoint. Now, it's how, depending whether or not you're using a volumetric or coulometric titrations, how the iodine is introduced. Volumetric titrations, you work with a standardized solution of iodine, drop. Iodine solutions are notoriously unstable in some applications that are UV, require constant re-standardization. That's why the coulometric titration is a little more convenient, where the iodine is actually generated in situ by a set of electrodes and then consumed as it reacts with the water. The reaction of the last slide, and then the first excess of iodine is detected by the electrode, signaling the endpoint. And if you go through the math, it's about 10.7 coulombs per milligram of water. Interesting. And coulomb is a measurement of current over a period of time. So the amount of current is measured by the microprocessor and then used in the calculations to determine how much water is present in your sample. That's questions, comments, Greg? Yeah.